Good morning, everyone. My name is Charles Sawyers from Sloan Kettering. I'm delighted to welcome you to this special edition of MSK Science Spotlight, and where we're going to honor the recipients of the 2020 Breakout Prizes. Before I start, though, I want to, we all recognize and care deeply about the events going on in the world and our community today. But for the next hour, we're going to recognize and celebrate the achievements of three exceptional postdoctoral fellows across our Tri-I community who are today's recipients. Before I introduce the first presenter, I want to give you a little history of the prize. About 7 years ago, Corey Bargman from Rockefeller, Lou Cantley from Weill Cornell, and I each independently received a phone call notifying us that we were each recipients of a new award called the Breakthrough Prize in Life Sciences. And it came with a rather substantial cash prize. So after recovering from the shock of our unexpected good fortune, the three of us hatched a plan to pay it forward by creating a new award for outstanding postdoctoral fellows. And we had three primary goals. The first is to create a prize with a scaled down but substantial cash component. The second was we wanted to give multiple awards each year across the tri-institutional community, and the awardees would be selected by a joint selection committee. And just the process of going through this, we thought would promote further interaction and collegiality across our three institutions. And finally, we wanted to endow the prize so that it could be given in perpetuity. So each of us made initial contributions to get it started. And then we sort of challenged our leaders and trustees at our three institutions, all of whom immediately embraced the idea. And so we have established that this is a perpetual prize. And it allows us to give several $25,000 cash prizes each year across the three institutions. For this year, I especially want to thank Tim Ryan at Weill Cornell, who chaired this year's selection committee, as well as the other committee members, Lee Gahn and Tim McGraw, also from Weill Cornell, Sasha Rudinsky, Chris Lima, and Maria Jason at MSK, and Luciano Marafini, Sahel Tabazoy, and Leslie Bosahal at Rockefeller. So let's get started. So here's today's three recipients. At the bottom, you see an email address or a tweet address in which you can send questions. The questions will be handled at the end of the three presentations. So please send them in as we go through, and then I'll moderate a Q&A session at the end. So without further ado, I'd like to introduce our first winner, Dr. Manu Sethi. Welcome, Manu. Congratulations.
Thank you.
So Manu obtained his undergraduate training in India, where he majored in computer science and engineering. Then he came to the United States about 12 years ago, first to do a master's in computer science at Columbia. Then he did a PhD in biophysics and systems biology at Weill Cornell. And his thesis advisor was our own Christina Leslie at MSK, where he worked on computational modeling of gene regulatory programs in disease. Then after completing his PhD, he joined Donna Pear's lab as a postdoctoral fellow about 6 years ago, where he has absolutely been on a tear, developing innovative new computational methods to address really critical questions in biology. And he's developed at least three important algorithms that are widely used now. One is to model bifurcating single cell trajectories. Another is to model the continuities in cell state transitions and fate choices. And the third is to harmonize single cell RNA sequencing data sets at discrete time points. In addition to these talents on the computational side, he's very clever at coming up with catchy names for these algorithms, which go by names such as Wishbone, Palantir, and Harmony. If you get a chance to look at his CV, you'll see a very long list of seminal publications, both as lead author and as a key collaborator with many of the leading cancer biology labs at MSK and elsewhere across a wide variety of topics. And I, personally have been lucky enough to work with Manu on one such project involving lineage plasticity of prostate cancer cells. Of course, Manu's talent has not gone unnoticed. As a graduate student, he received the prestigious Horsfall Fellowship, and now today he receives the prestigious Breakout Prize. And I also see that he's been recruited to an independent faculty position at the Fred Hutchinson Cancer Center in Seattle, where he will soon be setting up his own group. But today he'll tell us about his most recent publication on embryonic development of the gut tube. And I will now hand it over to Manu. Congratulations.
Thank you, Charles. Thank you so much for the kind introduction. I want to begin by thanking the Breakout Prize founders for establishing this amazing award. I'm deeply honored and really thrilled to receive this award. And today, I would like to talk about my postdoctoral work in characterizing lineage decisions in development trajectories using single-cell data. Next slide, please. As Charles mentioned, my primary research interest is to understand how a cell decides its lineage fate. And the primary approach I take to address this problem is to develop algorithms that model these developmental trajectories using the recent and exciting advances in single-cell genomics data. Next slide, please. And as Charles mentioned, one of the recent algorithms I developed is called Palantir. Palantir is a novel trajectory detection algorithm which models continuities in not only cell state transitions, but also cell fate choices. And Palantir accomplishes these continuities in cell fate choices by computing for each cell what's the probability of that particular cell reaching all of the different terminal states. And next, I would like to give a very brief technical introduction into how Palantir works. Next slide, please. Yeah. We start with constructing a nearest neighbor graph to approximate the phenotypic landscape on which the differentiating cells lie. We then determine a pseudo-time order by using shortest path distances from the predefined start cell, and the pseudo-time order effectively represents the developmental or differentiation progression in this trajectory. And finally, we combine the nearest neighbor graph and the pseudo-time order to model the developmental trajectories as a Markov chain. And this forms the core idea of Palantir. Modeling these differentiation processes as a Markov chain allows us to not only determine the terminal states automatically in the state, but as I show in the next slide, it also allows us to compute probabilities or lineage probabilities for each cell. An added advantage of these lineage probabilities is that we can quantify the information content or using entropy of these probabilities, and that we nominate to represent the differentiation potential of the cell, a predicted measure of the plasticity. As an example, the trajectory here begins close to where I have marked cell 1. And as you can see, this cell has high probability of reaching any of the 3 terminal states, and naturally is accompanied by high differentiation potential, which gradually drops down as the cells differentiate to the different terminal states. We next applied the Palantir algorithm to characterize, next slide please, to characterize mouse embryonic development with a specific focus on the endodermal layer. And this was a fantastic collaboration with Sonia Novochin and Kat Hadjontonakis lab in the developmental biology program at SKI. We generated a large-scale single-cell RNA-seq dataset spanning the pre-implantation blastocyst stage to the mid-gestation stage of the mouse embryo. And we performed extensive analysis with this data. And today I would like to take this opportunity to describe 2 short vignettes of what we found about embryonic development. Next slide, please. So I want to start at the embryonic day 5.5. This is the first post-implantation stage we have of the embryo. And then the embryo proper is comprised of 2 cell types here. One, the inner epiblast cells shown in red, which is surrounded by the visceral endoderm cells shown in blue. Next slide, please. And when we look at the single cell RNA-seq data, this is a force-directed layout where on the left, the cells are colored by the measured time point, and on the right, the cells are colored by the cell times. And even just with this visualization, what was very surprising to us was that we see a stream of cells which seem to connect the epiblast in red to the primitive endoderm or the visceral endoderm in blue. And this was a very surprising observation because the differentiation of the epiblast and the primitive endoderm from the inner cell mass was known to occur 2 days prior to this stage, and with no evidence of possible transdifferentiation between these two cell types. Next slide, please. Therefore, to characterize this better, we applied Palantir. And without going into too much detail, what we found was we identified a small set of epiblast cells, specifically at the embryonic day 5.5, which showed an increased potential to differentiate towards the visceral endoderm. And this means we can make a very concrete prediction now that there is a transdifferentiation at the post-implantation stage, and the transdifferentiation is from the epiblast to the visceral endoderm and not in the other direction. And this, of course, is just a prediction, and of course would require extensive validation. Next slide, please. And Sonia Novach and our collaborator performed really extensive lineage tracing experiments using tree reporters and definitively prove that there is indeed a transdifferentiation, specifically at the post-implantation stages, and that the transdifferentiation is from the epiblast to the visceral endoderm and not in the other direction, providing a validation for a prediction we made with Palantir. And I really want to emphasize here that we were able to make this prediction and identify this small set because we used Palantir to model these cell fate choices in a continuous manner. Next slide. So next, I want to switch gears to the gut tube. The gut tube, shown in blue here, defines the anterior to posterior or the head to tail axis of any mammalian embryo. And despite comprising really less than 4% of the cells of the embryo, next slide, the gut tube forms the precursors of the cells which form all the major endodermal organs of the body, highlighting how central this is not only for axis definition, but also establishing the endodermal organs. We performed again single cell RNA-seq of the gut tube. Next slide. And what you see on the left is a nice ordering of cells, again a force-directed layout of the gut tube cells, where there is a nice order from the anterior to the posterior. And this is, of course, very, very qualitative, but therefore we wanted a quantitative measure of the spatial organization of cells. Therefore, we were able to use the pseudo-time principles, which we use in Palantir, to define a very robust and accurate pseudo-spatial ordering of the cells along the gut tube, spanning the anterior to the posterior. Of course, the next question was, what exactly is in the what exactly are the cells doing or representing in the pseudo space order? Next slide. Therefore, we clustered the data. That's shown on the left, and on the middle, you see that the clusters do really line up very well along the pseudo spatial axis. To annotate these clusters, we identified some differentially expressed genes, and we were very excited to see that the order in which we annotate the clusters are exactly the order in which the organs develop in the developing embryo. And this means we can conclude that the cells in the gut tube are organized into these spatial territories of organ precursors. And this is exciting because if you look at them under the microscope, the cells of the gut tube are morphologically practically near identical. But here we can show that at the transcriptional state, the cells are primed for the ensuing organogenesis prior to any morphological changes. Next, we wanted to investigate what are the determinants or what are the genes that establish the pseudospatial ordering. Next slide. And of course, naturally, we looked at the Hox genes, and as you can see, the Hox genes, at least in the gut tube, are not at all informative to define the spatial organization of the cells at this stage. Instead, we developed a regression model And through that, we were able to identify a core set of about 20 transcriptional regulators which can near perfectly recover the pseudospatial ordering of the cells, highlighting how important the transcriptional priming is a result of the transcriptional regulation. And naturally, given the extremely central nature of transcription regulation, our next step was to generate single cell ATAC-seq data. Next slide. And what I'm showing you here is initial results from our single cell ATAC-seq data. At the top panel is the RNA-seq representing the organ territories. And in the bottom, what you see is single cell ATAC-seq, where we were able to recover both the organ territories and the pseudospatial ordering independently of the RNA. We are still in the early stages of analysis, but I wanted to give you a sneak peek. Next slide. And just an example of in the direction where we are heading at. In the middle, what you see is, again, a force-directed layout highlighting the expression of HOXC9, which is specifically expressed in the posterior. On the right, what you see is epigenomic tracks or epigenomic landscape derived for these organ territories using the single-cell ATAC-seq data. And the peaks or the enhancers that are highlighted, as you can see, are very specific to the posterior territory, and we speculate that these are the enhancers that are establishing this expression pattern. And despite this very focused and local expression, what you see also is between these two, let's say, posterior specific peaks, there is some other well-established enhancers which seem to be common across across the gut, highlighting importance of plasticity. And then final point I wanted to make here, at one of these posterior specific enhancers, we did preliminary motif analysis and found that it's predicted to be bound by CDX2, a well-known posterior regulator. And this means we can really start piecing together the mechanisms of how these expression patterns tend to be established, and then that's where we are going towards in terms of our methods development next. Next slide. And I want to finish by thanking all the people who made this work possible. First and foremost, I want to thank my postdoctoral advisor, Dana Peer. Dana has been an incredible mentor to me, and she has been a constant source of guidance and inspiration, and I'm very grateful for her. I want to thank Kat Hudson-Tonakis and Sonia Novochin in Kat's lab for a really stimulating and a fun, joyous collaboration on the Endodome project and many ongoing projects. I want to thank many members, past and present, of the Peer Lab, who made it a very fun and stimulating environment to perform research in. I want to finish by thanking the Breakout Prize founders and the prize committee for conferring me this award, and thank you very much for your attention.
Thank you, Manu. That was absolutely terrific. I have several questions, but we're going to wait till the end. Just to remind you, all of you who are watching, and if you happen to join late, we're going to take all the questions at the end. So if you have them, please email them to oset@mskcc.org, or you can tweet to @mskeducation. But Manu, what I want to do now is advance the slide to a tribute from Dana to you. So I'm now going to play that video.
Hey Manu, congratulations on your well-deserved Tri-Institutional Breakout Prize. You are a unique and gifted scientist, driven by curiosity and passion, and empowered by unique interdisciplinary gifts. You're an artist. You can actually see the data, see the structures and the patterns within, just by playing with it, exploring it. You intuitively see the structure in the data. Yet you have the rigor and the skills and the understanding of how to take what you intuitively see and rigorously formulate it into statistical tests, into powerful algorithms. And you're not limited to simplistic algorithms. Your software engineering skills really allows you to engineer very sophisticated algorithms that will run fast enough on huge data sets. And finally, you have the biological depth and understanding to understand what is important, what is exciting, what is new. Taking all that together, I cannot wait to see what you'll achieve as an independent PI and all the findings and breakthroughs that you will make in the future. So again, a huge congratulations. I am so proud of you and proud to be your mentor.
Congratulations, Donna, that was so nice. Okay, so thank you, Manu. I now have the pleasure of passing the baton to my friend and colleague, Corey Bargman, who's going to introduce the next winner.
Good morning, everyone, and welcome. At a trying time for us as a city, as a country, as a community, we have the joy of taking a step back and celebrating the things that we care about the most right now in our own particular zone, which are the successes of science, both individually and globally. And here in the context of the Breakout Prize, it is such an honor to introduce Alex Meskhi from Luciano Morafini's lab, who is our next Breakout Prize winner. Alex did his PhD work with David Rudner at the Harvard Medical School. Now, we share our planet with many, many prokaryotic organisms, and they have been around for much longer than we have, and they are incredibly diverse and variable. And it's easy for us to think that we understand bacteria because they are small and simple, but in fact, they are complex, incredible, and successful. And by studying more of them and by thinking more about them, we can learn things about the world that we did not know. Alex showed that in a very striking way by discovering in his graduate work an entirely new pathway for cell wall biosynthesis that had biochemically not been recognized in the bacterium Bacillus subtilis, a remarkable new insight into the biology of these organisms. And then as a postdoc with Luciano at the Rockefeller University, where he came 4 years ago, he has taught us even more about the bacterial world. He has been studying the CRISPR-Cas systems of bacteria. Now, many scientists are fascinated by those for our own reasons, because those are tools that we can use in our experiments. But the Morafini lab, and Alex beautifully exemplifies this, has instead been studying them in their natural context to understand what they mean for the bacteria. And again, taking this idea of thinking about the bacteria and their diversity, Alex has chosen to study not the well-characterized Cas9 system, but the type 6 Cases, the Cas13 system. It was known that this kind of enzyme targeted RNA rather than DNA. We know that the CRISPR-Cas systems in bacteria are being used to fight off phages and other parasitic DNA elements. And so it made sense to think that they might be involved in fighting off RNA phages. But Alex discovered, amazingly, that instead they fight off DNA agents and DNA phages through an astonishing mechanism in which the bacterium actually shuts off its own RNAs, its own growth, and ultimately dies in order to protect other bacteria within the colony. This is an unexpected and remarkable finding that he will tell you about further today. Alexander's work is characterized by creativity, conceptual insight, and experimental precision. It is an honor to hear his work here at the Breakout Prize Symposium.
Okay, thank you so much for the introduction, Corey. And I'd really like to thank all of the Breakout Prize Selection Committee and Professors Bargman, Cantley, and Sawyers for this incredible honor. I'm really excited to tell you all today about my postdoctoral work in Luciano's lab, which was focused on how the RNA targeting CRISPR-Cas13 system protects its bacterial host from infection by viruses, aka bacteriophages. So if you can advance the slide. In nature, CRISPR-Cas systems, as Corey mentioned, are immune systems that protect bacteria from infection by phages, and they do that by using RNA-guided nucleases like Cas9 to cleave viral nucleic acids. A typical CRISPR locus looks like the one shown on the left, with short repeating DNA sequences shown in white, and unique spacer sequences between them shown in colors, with CRISPR-associated Cas genes next door. In the first phase of CRISPR immunity, spacers are required from the genome of an infecting phage and integrated into the CRISPR array, where they're stored as memories for subsequent infections. Then to achieve immunity, the spacers are transcribed and processed into small guide RNAs, which associate with Cas effector nucleases to find and cleave matching sequences called protospacers in the viral DNA. And that's a generalized overview of how CRISPR systems work, but as Corey was alluding to, CRISPR systems are much more diverse than most people appreciate. Bioinformatic efforts over the past decade have identified thousands of CRISPR systems across bacteria and archaea, and they're broadly categorized into 6 types, which are unrelated to each other in sequence and structure. And while most of the CRISPR types ultimately result in cleavage of the viral DNA, the type 6 CRISPR system exclusively cleaves RNA targets. And so when type 6 CRISPR systems were first discovered, elegant work from Feng Zhang's lab at the Broad Institute found that they encode a single effector nuclease called Cas13, which associates with a guide RNA and binds complementary RNA protospacer shown in red on the bottom right. Once Cas13 binds to an RNA protospacer, it's licensed for both on-target cleavage of the bound protospacer, we call this cis-RNase activity, but it's also allosterically activated as a non-specific RNase. So it will cleave any RNA in solution, and we call this trans-RNase activity. This was observed in vitro in the initial study exploring Cas13 biology. On the next slide, we had questions about how Cas13's RNase activities interfere with phage infection. Unlike eukaryotic viruses, the vast majority of phages that we know of are not RNA phages, but have a DNA genome. And so we wondered whether the RNA cleavage activities of Cas13 could interfere with infection by a DNA phage. And if so, what are the relative roles of the on-target and off-target cleavage activities? So Cas13 is not present in any model organisms like E. coli or Bacillus subtilis. And instead, I developed a genetic toolkit for modifying one of its natural hosts, Listeria saligeri. And I found that when programmed appropriately, Cas13 in Listeria saligeri could interfere with transformation by a targeted plasmid, shown on the top right, or interfere with infection by several different DNA phages, shown on the bottom right. On the next slide, one of the first, as part of characterizing this system, we wanted to understand which sequences could be cleaved by Cas13 and which could not. All CRISPR systems have strict requirements about the sequences that can be cleaved. For Cas9, it strictly requires a 3-nucleotide protospacer adjacent motif, or PAM, in the target DNA. And this plays a role in preventing autoimmunity. There is no PAM in the CRISPR locus itself, and this is why Cas9 does not cleave its host's genome. So for Cas13, in using Listeria saligeri as a model, I wanted to understand what the PAM was, and also how does it prevent autoimmunity. And I found that there is no specific conserved sequence like a PAM required for Cas13 cleavage of target RNA. Instead, what we found was that Cas13 senses the amount of complementarity between the guide RNA and the target RNA, and uses that to decide whether or not to make a cut in the target RNA. For a foreign target RNA from a phage, there's complementarity between the spacer region of the guide RNA, shown in orange, and the protospacer region of the target RNA. But that complementarity does not extend into the constant region of the guide RNA, which is shown in black. We did find that for targets that have extended complementarity into that region, they're not cleaved by Cas13, and they seem to be sensed as self-derived RNAs. And interestingly, 3 of the 6 CRISPR types exclusively cleave DNA targets, and they all have a PAM requirement for cleavage. But both the type 3 and type 6 CRISPR systems are able to cleave RNA targets, and they similarly lack a PAM requirement and have evolved a very similar complementarity sensing mechanism to avoid being accidentally triggered by self-derived RNA targets. On the next slide, we were interested in how Cas13's RNA cleavage activities ultimately interfere with phage infection. And I mentioned before that Cas13 is allosterically activated by target RNA to become a non-specific RNA cleaving enzyme. So we see this trans RNA cleavage activity in vitro. We found that that was true in vivo as well. So if we program Cas13 to target a DNA phage, then we not only, using RNA-seq, we not only detected cleavage of phage transcripts, but also cleavage of most of the host transcripts as well. And in the movies that we're going to play on the top right, we'll see the consequences of Cas13 cleavage for the infected hosts. So both of these movies are listeria cells bound by fluorescently labeled phages. So the green dots are phages. And if we play the movie on the left, we'll see that in the absence of CRISPR, the phages lyse the cells. Whereas in the movie on the right, these cells have Cas13-based immunity against the phage, and we can see that they go dormant. They stop growing, they don't lyse, but they also don't release any viral progeny, and as a result, the bacterial population is saved. And we found that Cas13's dormancy-inducing effects that make it a very robust immune system. Because CRISPR in general is a sequence-specific immune system, that means it can easily be overcome by escaper mutant phage that evolve mutations in the target site and can no longer be recognized in a sequence-specific manner. For Cas9, we see escaper mutants arise all the time, and they can sweep through the population. But for Cas13, we didn't see any escaper mutants, and we found that this is because when Cas13 is activated by a wild-type phage, its non-specific RNase activity neutralizes both the wild-type phage as well as any co-infecting escaper mutants. And so in this way, Cas13 is able to nip outbreaks of escaper mutants in the bud before they can take off. And then finally, on the next slide, we had our most recent project focused on how phage overcome Cas13 immunity, because this is an arms race, and of course, phage evolved a way to overcome Cas13 immunity. Many phage encode anti-CRISPR proteins, which are small proteins produced during phage infection that inhibit the enzymatic activities of Cas enzymes and allow the phage infection to succeed. These anti-CRISPR proteins had been discovered for 4 out of the 6 CRISPR types, but not for Cas13. I had the opportunity to supervise a very talented surf student last summer named Alice Castle, and Alice identified a phage, a listeria phage in our collection that could overcome Cas13 immunity. And she spent the whole summer narrowing down the gene responsible until she found a single gene, which we called ACR6A1, which is necessary and sufficient for inhibiting Cas13 in immunity against both phages and plasmids. And we were able to reconstitute ACR6A1's Cas13 inhibition activity in vitro. It blocks both the on-target and off-target RNA cleavage activities of Cas13. And then finally, in a really fun collaboration with Ning Jia, a postdoc in Dinshaw Patel's group at MSKCC, we solved the co-cryo-EM structure of ACR6A1 bound to Cas13. And ACR6A1 is shown in blue in this structure, and it binds to Cas13 in a way that it occludes the guide RNA and prevents it from base pairing with target RNA. And so on the next slide, I'd just like to thank my postdoctoral advisor, Luciano Marafini, who's been an amazingly supportive mentor, the Marafini Lab members who have made it really fun to come into work every day, and special thanks to Alice Castle, who is that surf student whose results initiated the anti-CRISPR project, Albina Kozlova, who is our star technician who has been carrying the torch ever since, and Ning Jia, who is our structural collaborator at Dinshaw Patel's group. And I would just like to take this final opportunity to thank the Breakout Prize Selection Committee once again. I'm thrilled to be selected for the prize. It's a great honor. I think the events of the last 2 weeks have challenged us all to find ways to combat injustice. And so I've decided to donate a portion of the prize money to the NAACP Legal Defense Fund. And with that, I thank you for your attention, and let's continue the symposium.
Thank you, Alex, for that wonderful presentation and for those inspiring remarks at the end about making a difference. We're now going to hear from Luciano Marafini, whose lab you worked in at the Rockefeller University.
Hey, Alex. I wanted to congratulate you on this tremendous honor that is the Breakout Prize, and very happy that you were selected. I think that you deserve it so much. For those of you who don't know, Alex came to my lab with a plan in mind. He decided to study these weird CRISPR-Cas systems that target RNA and make the cells very sick when they're infected by viruses. And Alex designed all the reagents before coming to the lab, so he hit the ground running, and that paid off because he had tremendous publications, very high quality, and also very, very quickly paced. And I'm not surprised that you were so successful, and I wish you the best in your next step of your career, which will be to start your own lab. I'm sure you're going to be as successful as you were in my lab, or even more. And I can't wait to see how well you do in the future. Congrats again.
Thank you, Luciano.
Thank you, Luciano. Congratulations again, Alex. And now it's my pleasure to introduce Louis Cantley, who will introduce our third and final Breakout Prize winner.
So I'm delighted to introduce Dr. Ana Patricia Gomez from Weill Cornell, the winner of the 2020 Breakout Prize from Weill Cornell. Anna's a postdoctoral fellow in John Bonis's laboratory working in the field of cancer biology. She served as a visiting student at Harvard Medical School with David Sinclair. And during that time, and in fact, throughout her career, even as a postdoc, she's really been interested in aging, and particularly the role of metabolism in aging. As a graduate student, she made the surprising discovery that there are changes in specific metabolic pathways prior to genetic damage during aging. And so this raised the possibility that one could intervene in these metabolic pathways that are deregulated and either reverse or slow down aging. In John Bonis's laboratory, she continued to look at metabolism and aging and epigenetic events, and made a very surprising observation of epigenetic mechanisms relying on histone H3 variants changing that led to progression with aging and also tumor progression and metastasis. So Anna has received a number of notable awards, including the K99ROO Pathway for Independence Award from the NCI, the 2017 Stat Wunderkind Award, and she won the Bench to Bedside Initiative Tri-Institute Shark Tank. She's currently a Susan G. Komen postdoctoral fellow, and she's published papers in all the top journals: Cell, Nature, Science, Nature Communications, etc. And I have had the pleasure actually of collaborating with her on several of her publications. So please join me in congratulating Anna on her scientific accomplishments and on winning this prize. And at this time, I'll turn it over to Anna.
Thank you, Lou, for your kind introduction. And I would like to start by thanking the Breakout Prize founders and the selection committee for selecting me. I'm very excited to be able to share with everyone today what I did for my postdoc. So as you may see from this slide, I have a long-lasting interest in understanding how aging regulates complex disease. Can I have the next slide, please? And so for my postdoc, I was very interested in the connections of aging and cancer, and that really stemmed from the observations highlighted here in this graph. So as we all know, aging is the main risk factor for cancer incidence, and that's highlighted here in the red line. But more than that, what I was really interested at the start of my postdoc was this observation here in the blue line, where we see that not only is aging main risk factor for cancer incidence, but also older patients are more likely to die of cancer, which when you think about it, you can think really in two ways. One being that as you get older, your body is more frail and therefore less able to withstand the harsh treatments that you need for cancer. Not only that, but you also have now a history of disease, very likely, which also affects cancer outcome. But I always thought that, well, aging means basically that every single cell in our body is different, which then yields an aged host a completely different environment for any disease, cancer included, which led me to think that perhaps there was a direct connection between aging and making a conducive environment for cancer progression, and then account for some of the mortality seen in older patients. So in the next slide, we'll see the approach we took. So modeling aging is rather complicated. And we decided at the time to take what I thought was a simple approach and just take some cancer cell lines in culture, mostly carcinoma cell lines, and replace the culture media with, instead of using animal-based serum in that media, we decided to replace it with human serum coming from either young or old donors. And I should say that these owners were otherwise healthy, so they did not have any diagnosed disease at time of collection. And so we just changed the serum, cultured the cell for a couple days, and as you will see in the next slide, when we looked at the cells, they actually looked very different morphologically speaking. So the cells cultured with young serum, they looked pretty much like the parental ones that were cultured with animal-based serum. But the cells cultured with old serum, they looked very different. They were much more elongated, spindle-like, much like they lost their cell-to-cell attachments. And so that was interesting to me because that was reminiscent of epithelial to mesenchymal transition. And if you can go to the next slide, you'll see that we were actually able to validate these observations in many different ways. Here you can just see some biochemical markers of epithelial to mesenchymal transition, where we really see downregulation of the epithelial marker ecadherin and a concomitant increase in mesenchymal markers such as fibronectin and vimentin. So really showing that there is something in the old serum that triggers epithelial to mesenchymal transition in cancer cells. But why did I find this interesting at the time? So if you can go to the next slide, you'll see that epithelial to mesenchymal transition, although it's a developmental program, it is hijacked by cancer cells, or at least some cancer cells, to endow the cells with the ability to leave the primary tumor, go into circulation, and then establish themselves at a distant location and form metastatic lesions, which account for the vast majority of cancer-associated mortality. And so what we decided to do was to really see whether the old serum was just inducing EMT or was also causing aggressive properties in cancer cells. And so next slide, please. And so we did a quite extensive characterization of this, but for sake of time, I'll just show you one experiment where we tested the ability of old serum to affect metastatic colonization. So here, basically, we just took a triple-negative breast cancer cell line that in of itself already has some metastatic properties, and we cultured them for 5 days with young and old serum. Then we put them in, we injected them in the tail vein of nude mice and waited for a while to see how that they would colonize the lungs. So at this time frame, which was about 6 weeks, we can see that cells that were cultured with young serum, they're starting to form some colonies in the lung, which is normal for this cell line in this time frame. But then when we looked at cells cultured with old serum, and these cells were just cultured for 5 days and haven't seen old serum for the next 6 weeks, we see that now they have a much heightened ability to form colonies in the lung, really highlighting how circulatory changes that occur with aging can modulate tumor progression. And if we go to the next slide, you will see that in a nice model that we put together. And so, and now next slide. So the question now is, what is it in the old serum that can produce such aggressive properties? And so we profiled the serum quite extensively, but to cut a very long story short, I will just dive into what we found. So on the next slide, you'll see that we narrow it down to the accumulation in the serum of a single metabolite called methylmalonic acid. You can see on the left panel the distribution of the old donors. There is a very high upregulation of methylmalonic acid in the serum of old donors. And so what methylmalonic acid is basically a byproduct of propionate metabolism. So in other words, carbon coming from brain chain amino acids or chain fatty acids fuel the TCA cycle through this propionate metabolism. And when there is too much carbon flowing through the pathway, or the downstream enzymes such as MCE and MUT or vitamin B12 deficiency occurs, there is an accumulation of methylmalonyl-CoA, which then gets transformed into methylmalonic acid that is excreted by the cells. And so, next slide, please. So again, we did quite an extensive characterization, but I'll just show you one experiment that I think really makes the point of how methylmalonic acid is such an important regulator of tumor progression. So we wanted to mimic what was occurring in old people in the serum in circulatory changes. And so to do that, we did sub-Q injections in immune-compromised mice every day to raise the levels of methylmalonic acid to levels similar to what we see in the old donors. And when we did that, we then implanted the cancer cells into the memory fat pad and waited for a while to see what happened. And as we starting seeing cancer progression, what we saw was that tumors that were implemented in mice that had high levels of circulatory methylmalonic acid actually grew much faster. As you can see in the image, the tumors are much, much bigger. They also starting spreading to other parts of the body, much more like than the cancers grown in vehicle mice, as you can see in the middle graph, and resulting in overall decreased or significant decrease in survival of these mice, as you can see in the right panel. And so together, this data really shows in the next slide that aging does have a direct role in tumor progression, in part by leading to the accumulation of methylmalonic acid in circulation. And we have actually identified how methylmalonic acid has these progressive properties, and that is through induction of TGF-beta signaling and SOX4, which is a transcription factor that is responsible for the progressive remodeling that we see in the cells, and therefore responsible for tumor progression when driven by methylmalonic acid. And so lastly, I just want to go to my acknowledgement slides and thank John Blanes, my postdoctoral mentor. He was fantastic, always very supportive. I cannot thank him enough. And several people in the Blanes lab without whom I wouldn't have been able to do so many of the projects that I did. All our collaborators, they were always fantastic, and my sources of funding. And once again, I would like to thank the Breakout Prize founders for their generosity, and thank you all for your attention. And if we can go to my next slide, I would just like to tell you that if you are listening and you are interested in understanding more how aging factors in cancer, just check out my lab at the Moffitt Cancer Center. We just started, and just contact me if you have any interest.
So thank you. That was really terrific. And I'm going to apply for your lab immediately. I have some ideas of experiments. It's anyway, that was terrific. Now we want to hear from John Blennis from Weill Cornell Medicine, and if you could play the video, John.
Anna, first, congratulations on being awarded the Tri-Institutional Breakout Prize for Young Investigators. You have been an amazing, thoughtful, and creative scientist both before and while you were in my lab. You have made groundbreaking discoveries at every level. You have been a fabulous collaborator and a truly incredible mentor to many students and postdocs. This award is well-deserved. So once again, I congratulate you, and we all look forward to your continued progress and more discoveries in your new lab.
Okay, so at this point, I'll turn it back to Charles, who will lead the question and answer.
Okay, so, wow, 3 great talks. I'm sure there are lots of questions. So I'll just leave this slide up for just a second so you can get the email address or the tweet address. Then I'm going to stop sharing. And I'd like everyone to come back on, our 3 award winners and our 3 announcers. And I'm going to go check the Q&A board. But while I do that, I'd suggest that Corey and Lou, you get first dibs on questions, and you can self-moderate while I go read the.
Corey, do you want to go first?
Sure. I have a question. I'm just I'm still kind of dazzled by the talks. I have a question for Alex, which is how many more of these kinds of CRISPR-Cas systems are there that we haven't discovered yet, or that we haven't characterized? How many new discoveries are there to make here?
Yeah, I think over the last 4 years, people have asked the bioinformaticians that have kind of driven the field forward, when is type 7 coming out? But it doesn't look like as far as from the existing sequence databases, there are any other large groups of CRISPR systems, barring things that we haven't sequenced yet, at large groups of bacteria or archaea that we just don't know about yet. But I really can't overstate the diversity of these systems. So there are 6 types of CRISPR system, which are completely unrelated, between the nucleases. Like there's no sequence similarity between Cas9 and Cas13. And then even the 6 types are broken into 33 subtypes. And even between the subtypes, there's very limited sequence similarity between a type 6A CRISPR system and a type 6B system. It would be like 5% amino acid identity or something. So I think even though there might not be new large groups coming out, we really haven't characterized most of these systems in detail, only a handful at the molecular level, and the vast majority of them have only been characterized in vitro or in E. coli or in mammalian cells, and almost never in their native bacterial hosts.
Alex, there's a follow-up kind of question from the ether. What about the potential of applying what you've done to RNA transcript editing? Is there any hope?
Well, yeah. So this isn't my work, but work from Feng Zhang's lab has they've coupled Cas13, a nuclease dead Cas13, to an ADAR domain, and they've achieved RNA editing in mammalian cells. I think that the efficacy depends on the cell type and the delivery and guide RNA design, but it does seem that they have multiple platforms for RNA editing based on Cas13, so it definitely is a reality.
Okay, thanks. Lou, do you have a question?
Yes, I have actually two related questions. And so the first is that we know that with aging, insulin resistance correlates very strongly. And we know in worms that age 1 is PI3 kinase, age 2 is the insulin receptor. The whole pathway is involved in aging, and that there's a global effect on in the worms, in that if you only affect a few cell types, you end up affecting the whole body, indicating that it's not, every cell is not aging independently, it's interactive. So the question you can see I'm leading up to is if you look at insulin resistance, that you see methylmalonate higher in patients with insulin resistance. And secondly, do you think that worms are generating methylmalonate when that pathway is on?
So it's definitely a possibility. I mean, PI3 kinase is central to aging, as you just pointed. Methylmalonic acid is definitely possible to be made in worms. It is conserved all the way to bacteria. So it is a very conserved pathway. On how methylmalonic acid may or not affect insulin resistance, I believe that it may. In fact, we see in our transcriptomic analysis a lot of insulin signaling coming up when we treat cells with methylmalonic acid.
Actually, I was suggesting the other way around. Insulin resistance is causing methylmalonic.
Yes, but we don't know that. I haven't tested, but there's definitely something I will in the future, even with the links with obesity and cancer as well. I mean, it makes sense. There is a lot of chain fatty acids can be fueling this pathway. So just by metabolic flux, it is a possibility that insulin resistance and obesity will lead to an increase in methylmalonic acid, which then will have all sorts of pleiotropic effects in multiple cells. But that's all new to be explored.
Anna, there's a question from the audience on MMA and whether, do you know, are there any effects on immunity or T cell function that might be related to what you're seeing?
Yeah, so that's actually an area that my lab is exploring now. There is definitely effect. We do have preliminary data showing that methylmalonic acid is actually a powerful inducer of T cell exhaustion. And so there is definitely something going on there that we are going to explore.
Well, that question actually comes from Larry Norton, someone you should get to know. He may be applying for your lab as well. Okay, Manu, a question for you. So you showed, you made that point early on about, you made predictions about the epi to VE transition that then you were able to prove through lineage tracing. But that lineage tracing benefited from the fact that there were some predetermined markers of those lineages. As we go forward, are there any, sort of technologies or other, tools that can help us, look at this on a greater scale? Because as you know, we're finding many more bifurcations in different aspects of adult biology.
Yeah, definitely. I mean, especially when it comes to experimental systems, I think there's really been an explosion of systems where you can do kind of parallel single-cell RNA-seq and lineage tracing using these CRISPR-based scratch map systems. And I mean, I think there, while there have been a huge number, there's still, in terms of the resolution, maybe they don't necessarily give us the complete picture, but I think there's been huge advances there. And that, of course, creates a major computational challenge as well. But I think as the resolution of these systems improve, we'll really get a much better picture. And essentially that gives us a ground truth of every lineage transition or lineage relationship that there probably is. Which in itself can serve as almost as a benchmarking experiments for computational modeling as well. But I think, yeah, combining the single-cell RNA-seq with the lineage tracing system can be extremely powerful when it comes to this and generalize without prior knowledge, let's say, of individual genes or markers.
And a follow-up question to that, you gave us that teaser at the end where you showed us the ATAC single-cell and integrating it with the RNA-seq single cell. But those are sort of two separate, samples that get analyzed. What is that going to look like going forward? I mean, are you happy with the way you sort of have to impute what's going on, from two different measurements in two separate cells and assume they're in the same cell, or where will we go with that?
Right. This is a lot of work that's happening now in the PAIR lab, particularly spearheaded by Zining Chu, who's a MD PhD student. One of the things we are really looking at is right. So can we do do a mapping between the RNA and ATAC-seq? Because the noise levels of these data sets are so high, we are thinking of abstracting them into some kind of metastable states, and that gives us a better way to map the cells as well. And I think, at least as our first pass, what analysis we have, when we do the mapping between the RNA and ATAC, we are very happy with how the results look like. And I think going forward, there's also there's quite a few technologies that are coming now where they're measuring RNA and ATAC effectively in the same cell. We'll see how that data looks like. I mean, at the very least, it can serve as, again, ground truth for our integration. But if that is something that is cost effective and gives us the resolution, then this kind of mapping question goes away. But if the resolution isn't sufficient, then we can continue to build very robust algorithms to map, which really gives us a much richer description of the system.
Okay, well, thank you. So we're actually at the top of the hour. I think this has been, well, at least for me, this has been a fantastic way to celebrate your achievements. And I'm so proud of all three of you, and we'll look forward to following your future success. And hopefully we can run into each other face to face sometime soon. So congratulations. And thank you all for watching. And have a great rest of the day.
Thank you.
Bye.